Nebraska, but he's at the Department of Food Science and Technology. And he's engaged in research that is very relevant to this partnership. He's interested in allergy, and he is the person who maintains Allergy online, the database. This is one of his major activities. He also does a lot of other work as well, and advises governments around the world. He spends a lot of time, I know, in India, other places. Richard is going to talk to us today about evaluating the potential allergenicity of GMOs intended for food use. And he's promised me that each of his slides is only going to have 1 point on it.
I did? Oh my God.
Oh,
can I remake it?
I think so. No, we were having a drink last night, and we agreed this, and he may well not realize that he signed a piece of paper to this place.
Okay.
Single point slides.
Okay, that's going to be tough. I mean, I've never been known to not have too many things to say.
I've seen some slides before.
Yeah, you have. You may see some again. Sorry. As TJ Higgins is a great collaborator, and actually the collaboration with TJ started, which by the way doesn't involve any exchange of money. It's just in exchange of energy and time. I had a student who's from Ghana who was interested in a PhD program with me. She'd been working on his transgenic pea a bit when she was in Ghana and did a little stint at Champaign-Urbana. She asked me to come, and she was working on pepsin digestibility. She came to my lab, did a PhD, stayed for 1 year on a postdoc. And because of that interaction, then I met TJ in Senegal a few years ago, and we continued to do some work. So it is a great pleasure to be here. Thank you very much to Michelle for organizing this, and for you for being here. I would like to acknowledge the fact, before I forget, that I do have some money from the U.S. Environmental Protection Agency for developing some methods for IgE binding and also for some basophil work. And the Food Allergy Research and Resource Program, I partner with Steve Taylor, and he provides a lot of support, and it's an important thing. I'm going to go quickly. I hope you don't mind. If you want, I will give you a PDF of these slides afterwards. That's no problem. I would like to today cover many things. Allergy, what is it? Because a lot of people don't understand what food allergy is. They may understand airway allergy, not food. What are the risks, not only of GM, but of foods? Because the real risk that's known is of everyday food that's not GM, except GM by nature. Nature has made many natural recombinations. Methods that are used for the allergenicity assessment, examples of the assessment including alpha amylase in legumes from TJ, also a little bit on Pry1, and finally just a teeny bit at the end about endogenous allergenicity assessment on soybeans. We've done a number of studies, and I'll talk a little bit about that, and is that a relevant question? And then I would like to leave mostly with the question, is post-market monitoring possibly relevant to consider for allergenicity of GM foods? I hope you all realize that historically we have learned over many, many, many eons what to eat, how to eat it, and what is safe and how to prepare it. But we also have to realize that wheat, while it's good for most people, causes celiac disease in a number of people, and once they're clear that they have celiac, they have to avoid wheat. Legumes, many but not all of the legumes, have to be cooked to inactivate lectins and trypsin inhibitors. And for most foods, people can eat them, but those with allergies have to avoid them, or they have serious consequences. If you're going to do assessments, they really need to be scientifically sound. The standard must be there, and the idea is that GM foods are as safe as the conventional foods that are there. You can never prove 100% safety. That is impossible. I'm not going to talk about all this, but please, if you do not already understand it, evaluating safety and utility of GM products is very complicated, and there's a lot of work that goes into it before it ever gets on the market. We're going to talk about allergenicity, but there are many other things relevant to PMM. I would say food allergens, well, allergens are proteins almost always. They are proteins. And Those are things that are not hazardous for most people, but only for those who are sensitized and actually allergic, not just have IgE. They are only hazardous for those who have specific IgE and also reactivity. Celiac disease is an enteropathy. People who are genetically predisposed and have celiac disease have to avoid those proteins or they have severe symptoms. Proteins are introduced as GMOs and they have to be assessed for potential risks of allergy. And if the gene was from a wheat relative, they also have to be assessed for potential celiac disease on a case-by-case basis. The natural history of food allergy, and I just came from the American Academy meetings in Orlando. There's great debate, there's great knowledge. I'll be at the European meetings in Geneva in June. There's a huge amount of knowledge. The reactions are reproducible. In the same person, same food, you get same or similar reactions. It may progress over time from general dermatitis and hives to vomit, wheeze, asthma, and anaphylaxis. While young children are more often allergic, allergies to cow's milk, eggs, wheat, and soy often resolves. People become tolerant. But allergy to peanut, nuts, and seafood is typically permanent. Celiac disease, for those with celiac disease, is permanent. A lot of things to think about. It's an immune response. It's complicated. It's not something that's easy to figure out. Allergy is really, that we're concerned about generally, is IgE mediated. It's a specific antibody class. You can have other antibodies to the same proteins. For celiac disease, you have to worry about T-cell responses also. Sometimes food allergy just causes runny nose, a little itch in the mouth, but sometimes for some people it causes much more severe reactions, usually in 10 or 15 minutes of exposure. Angioedema. Oh, you can get hives that will progress to eczema for some people, and that's a mixed T-cell and IgE response. You can have somebody go to anaphylaxis within 15 minutes, and if you don't have intervention with epinephrine and possibly some other things, they could die. This girl had the reaction due to a challenge because they thought she had outgrown her allergy, but it was done in a clinical setting. She did recover from this, but it was almost fatal. And if it was out in the real world, it could have been fatal. Celiac disease, which I'm going to spend less time on, it is a T-cell mediated event. You have to have the right MHC class 2, DQ, or D8. This, however, is prevalent at about 20 to 25% of the population worldwide, but yet 1 to 1.3% of people have celiac disease. So other factors play a role. What is celiac disease? It's a T-cell mediated event that makes your small intestinal villi disappear when you're exposed to wheat or wheat-relative proteins. You have a tremendous inflammatory response, and obviously you will not be nutritionally sufficient. Sensitization. You have to be sensitized. Immunity doesn't happen once, and you're always going to be reacting. It takes some priming and a reaction. It can happen at any age. Most commonly, food allergy is less than 3 years of age. However, it also happens when you're between 3 and 20, and rarely in older people. Quite often, you get a new food allergy when you eat something new. If an American goes to Greece or India or China, they're exposed to very different foods. They may get different sensitivities. People who are of Indian descent, grew up in India, come to the U.S., typically they get U.S.-type food allergies at some low prevalence. U.S. population, 300 million people, 30% have some airway disease, allergy, could be rhinitis, could be asthma, but only 2 to 4% of adults, 4 to 8% of young children have food allergy. So that's a real reduction. Severe reactions are relatively rare. Estimates in the U.S., 120,000 hospital emergency room visits per year, and less than 200 deaths. Most of them are due to tree nuts or peanuts. A few to eggs and milk. 8 foods in the U.S. account for 90% of severe reactions, and those are the reactive foods. Remember, these foods are made up of thousands of proteins. It's not just a single protein. In the EU, they add celery root, mustard seeds, sesame seeds, and lupin as important food allergens, so they have a little bit longer list. In India, we're not sure, but we think black gram, pigeon pea, mung bean, lentil, Bengal gram, all legumes, and eaten as a major protein source, are probably the major allergens. However, India, the prevalence of allergy in India seems rare. If you look in the US population, this is from Sampson's group, young children, prevalence, adults, prevalence, you see shifts because these people become tolerant. You see other phenomena because in the US, most kids don't start eating fish until they're older, shellfish like shrimp until they're older. Here's the prevalence data approximately. This is just a brief introduction. Again, I'll give you PDFs. Probably 50 deaths or more per year in the US, 3 to 5 major allergenic proteins, 5 to 7 minor allergens out of 10,000 to 40,000 genes. Soybeans, 3 to 5 moderate allergens, a number of other minor allergens, maize or corn, no published cases of fatal reactions that I'm aware of. One major allergenic protein in food, lipid transfer protein, 4 to 5 minor allergens. Very rare. So what the heck does IgE bind to? It's an antibody. It binds to some epitope on proteins. For many people, their IgE antibodies will recognize conformational epitopes if you cook the food or you chew it up a little bit with proteases. Many times this is destroyed and they would not have IgE binding anymore. Some people have IgE that binds to sequential epitopes, and there it doesn't matter if you cook it, it doesn't matter if it's chewed up a bit, as long as there's at least 2 epitopes hooked together, a person could react. Yesterday you heard TJ talk about asparagine-linked glycans, and he showed that there are some on alpha amylase inhibitor. A number of proteins have those. Plants glycosylate a little differently than humans do. There's variation in it. Out of about 1,200 different structures, a handful of them are known to bind IgE to some people. However, almost nobody has been shown to have any reactivity at all due to just IgE binding to glycans, and I'll talk about that later. If we assume I was sensitized to peanut, I had IgE against it, and then sometime later I start eating it, I chew the peanut up, I release proteins. If my IgE binds to a protein like ARA-H1 and it binds with 2 binding sites, it will induce a signal on mast cells and basophils, either in my mucosal surface or in my blood. And if you get 100 of these going on the cell, it releases histamines and leukotrienes, and within 10 to 20 minutes, I can have symptoms. Depending on my sensitivity, they may be severe, could be massive, I could end up dead if I don't get treated. Most people would have minor symptoms and it would be more localized. And in a little bit, if you're not severely infected or if you get treatment, within a day you're okay. People who are allergic must avoid the proteins that cause disease. That means avoid the whole source of the protein, like peanuts. It means reading labels on processed foods. It means also avoiding the ingredients that contain the protein. So if we transfer the protein as a novel ingredient into a new food or through GM, then you're at risk. But in reality, most of the risk for food allergy comes when the allergic person eats prepared food from a restaurant or from their friends, or from packaged foods or drinks that are not labeled or they didn't read the label. It is important to realize that one of the real safety assessment concerns for GM is that you want to make sure you do not transfer a protein that is already an allergen for this person or that person, or is not so similar to an allergen that it would cause a cross-reaction in one of those people, just like the real allergen. The history of the development of much of the safety assessment really comes down to DuPont, Pioneer at the time, wanted to improve the nutritional ability of soybeans, and they said, you know what, they're a little bit deficient in methionine. There are proteins out there that have high methionine. One of them is a 2S-albumin from Brazil nut. Nobody knew at the time it was an allergen. Steve Taylor talked to people in Pioneer and said, you know, if you're going to do this, realize that Brazil nut is an allergenic food for some people, so let's test it. They said, okay, so they supplied the material, they set up a good design study, and here's just a stain gel of markers, soybean, GM soybean, Brazil nut, and purified Burr E1. We now know it's an allergen. Here's an immunoblot of the same type gel, and they used antibodies, and indeed, the GM soybean has an IgE reactive band that is the same as in the Brazil nut, and it's the same as the purified protein. When they did RAST inhibition, they could inhibit IgE binding to the Brazil nut or to Burr E1 with the GM soybean, and the inhibition was very similar to the Brazil nut. If you use non-GM soybean, there was no inhibition, indicating the reaction was very specific to the Brazil nut. When they did skin prick tests with this and they looked at transgenic soybean and Brazil nut, they got similar reactivities. If they used non-transgenic soybean, there was no wheal and flare response in the skin of allergic subjects. Pioneer did the appropriate, responsible thing. They said, well, we're not even going to bother take the time to submit it to the regulators because it's clear there was a risk. And it never went on the market. People were not exposed to it through the food. The methods used to analyze this, it was clear that if you have a reason to believe that you might have IgE reactivity, you need to test it, and you need to test it very well. I was at the Codex 2001 meeting that developed the Codex 2003 guidelines, and that became the standard really for EFSA, for the US, for Japan, and for the allergenicity assessment. I've put in my perception of the risk, but this is the ranking anyway. The way it's done is, the first question, does the gene encode a protein that's known to be an allergen or induce celiac disease? And that is in a different segment, so sometimes people miss the celiac part, but it's in another section of Codex. And if you have a reason, if you took the gene from an allergenic source, you probably have to do serum IgE tests. Is the sequence of the protein sufficiently similar to an allergen to believe there could be cross-reactivity? Then again, you would have to do very specific IgE tests. Or for celiac disease, you may have to do PBMC challenge. The next question is, okay, there's no evidence it's an allergen from the other two questions, so then what do? Well, you look at things like protein digestibility, pepsin digestibility, and abundance in the food, and that may be an indication that it might be sensitizing de novo, or that if somebody is sensitized, that it would cause hazard, it would be a hazard and it could cause an allergic response. Finally, the question, and I used to believe more fully in this question, but I don't anymore, did insertion of the gene increase endogenous allergenicity? So you put the gene into soybean, did you boost the amount of endogenous allergens that are already there? Really, when you think about it, there's variation in normal soybean varieties. Is this worth testing? Unless you put in a transcription factor that's going to boost specifically the allergens, I really think it's not an important question, but it's one that we're kind of stuck with for the highly allergenic foods like soybean, and I'll talk about why that's hard to test. Because some people seem to not respond to the Word document very well, and they seem to want some kind of a flowchart. In 2008, we published this diagram and said, let's think about this. What does the weight of evidence from Codex really mean? Source of the gene, no reports of allergy, commonly allergenic, but there's a lot of shades of gray. Many things have a little evidence that a few people are allergic to. You have to be able to interpret that. If you look at the protein sequence to consider potential cross-reactivity, how identical does it have to be to an allergen to believe it would cause cross-reactivity? There are a lot of shades of gray. It seems that a very conservative 35% identity over 80 amino acids may have a little potential sign of a risk. If it's greater than 70% identity to a clear proven allergen, there's a very high chance that it's cross-reactive. But there's a lot of shades of gray. If either of these are on the right-hand side of the gray, then probably you have to do specific serum tests. And again, just like in diagnosis of allergy, there are shades of gray. A serum IgE test alone will not prove allergy, but it's a pretty good indication you may have risk and you may have to evaluate it further. So this is an important thing. It takes a lot of care. It takes patient selection. In terms of pepsin stability, there are arguments about this. Oh, the EU is considering, well, you need more physiological digestive forms than the simple test tube assay of pH 1.2 and 2. Guess what? 1.2 to 2 with pepsin in a fixed diagram gives you good predictive value about digestibility. If you go to pH 3.5, pepsin's almost inactive. If you go to pH 7, pepsin's dead. What's the relevance of that test? What's the relevance? Because some people take an antacid or whatever, it stops their acid. Does that mean they instantly become allergic? No. So let's stick to the assay that's predictive. If the gene is from a major allergen, and that would be defined, here's some examples, then you would have to do serum testing. And if it's not, then you probably cannot find people at risk, and you also probably have a hard time finding test subjects to look at. We all understand the principles. Gene sequence dictates RNA sequence, dictates amino acid sequence, dictates endogenous activity of the protein, and also potential allergenicity. So Cry1 works because of this. You don't get some magic out here. Questions to answer about bioinformatics? Well, you're looking, we would love to, and Heimo and others here in Austria do a great job of looking at 3D predictions, but we're not there where we can really 3D predict all allergens and understand the relationship. So a simple sequence, amino acid sequence test is good. And the questions you're answering is, are the proteins already known to be allergenic, or are they so similar that you suspect cross-reactivity? And if so, you have to do specific serum testing. It's absolutely essential that you use sequences that you are pretty confident are allergens. So we have sponsorship, and we're now at version 12, was released in February. That's a pretty good source, I would argue. And you also have to have some criteria, and these are not absolute, but you have to have some criteria, predictivity. And again, the question isn't, is it an allergen? If it's an allergen, why would you bother? If it's anything less than a clear allergen, you would have to think about whether or not you have to do serum testing in order to get it on the market. Here's the database. Here's the website. Right now, we're version 12, 1,600 proteins, 603 taxonomic protein groups. We also have sort of an experimental link database for celiac disease. This is not as well evaluated as our expert panel reviewed allergens, and I can talk about that later, but it was from my lab, and we think that we have a pretty good way to rule out the potential value or the potential risk from many genes that might be taken from wheat family proteins. If you're not taking a gene from a wheat family protein, there's no reason to believe that you should have to do this. So what are the major allergenic food sources? We consider and put in allergen online those that are food allergens, airway allergens, venoms, and salivary proteins. And I'm not going to go in detail. Realize that peanuts, big allergens for foods. Out of 10,000 proteins have a small number that are allergens. Soybeans, similar number here, but fewer proteins and fewer risks. Maize, similar thing, and fewer. Okay. What are the sequence evaluations? FAST-A gives you the best prediction, and it's more than 50% identical with a very small E-score. Then it's good to think it might be cross-reactive. To be cautious, a window of 80 amino acids. If it's greater than 35%, there's some possibility. If you get positive matches, you probably have to do serum testing or you will go nowhere in the regulatory arena. This is just to give a quick example. RH1, a well-known peanut allergen. If you do sequence comparison with allergen online, you get identity matches to itself at 100%, to pea at 65, lentil 64, soybean 63, lupin 62, tree nuts less. These, there's very little evidence for cross-reactivity. So the idea of 50% identity is pretty good. And if you look at short amino acid matches, then that's a higher number, but less reliable. So now let's get to the examples with GM cowpeas. And TJ did not talk about this, but Cry1 in cowpea gives you lepidopteran resistance. And Cry1 is a pretty well-known protein, and it's not known to be allergenic. It's highly digestible. It's from Bacillus thuringiensis. It's not known to be an allergenic source. So you should be able to move forward with that product quite rapidly with very little extra testing. Alpha amylase inhibitor is one of the big topics of research here. And as for Coleoptera res, the genes is from Phaseolus vulgaris, common bean. How many people eat common beans? 2, 3, 4, 5, 6. Oh, everybody here probably eats common beans. That means green beans, kidney beans, navy beans, pinto beans. Wow, there's a lot. And they're good sources for many people. And hey, there's very little allergy to common beans. There are some cases, but very few. What else do we know? Well, we know that alpha amylase has never been submitted for food approval, so there's potential hazard, a hard thing for TJ to get it through, but maybe it's okay. It's got an excellent history of safe use in common beans. There are, however, significant regulatory hurdles, but I believe from the data we have that it's probably safe and very low possibility of allergenicity, and we're going to talk about that. Again, Cry1, there's probably no reason to worry. Alpha amylase inhibitor. Okay, well, there are multiple asparagine-linked glycans on there. TJ talked about that yesterday. The protein sequence, we ran that against Allergen Online, and you're above the threshold of 35% identity to one protein. That one protein is peanut agglutinin. That is a protein that's in Allergen Online. There are a few published reports that suggest it might be an allergen, but really those reports are pretty poor. And those are reports really of this person's got peanut allergy. They have some IgE binding to it, but there was never a test to say, okay, we're going to take you and challenge you with peanut agglutinin and you're going to react or not. There aren't that kind of data, but there's little evidence. But anyway, we've done serum testing. The protein is pretty stable at pH 2. It is an abundant protein. You say, oh God, these are going to be problems for regulators. Well, they may be, but the overriding fact is how many people eat Phaseolus vulgaris? Oh, everybody. And how many people are allergic? Very few. What are the allergens? We're not sure, but we think they may be the homologs of ARA-H1 and a few other proteins like that. So here's the sequence comparison, and this was actually done on an earlier version of Allergen Online, but matched to peanut agglutinin, 45% over 80, 35% overall, and we're doing serum testing. Again, there was very few reports. We've used 34 peanut allergic subject sera, and only one had significant IgE binding to peanut agglutinin. Okay? Oh, there were 5 weak binders. We've done extensive tests with inhibition to look at that, and actually it's not cross-reactive because of the similarity to peanut agglutinin. However, in doing the tests, sometimes I start forgetting where I am. You have to do, I'm going to talk a little bit about how you do serum IgE tests, and then we'll go to the results. It's absolutely essential that your test be shown to be specific, requires some validation. You need to have positive and negative controls. You can't just put on alpha amylase inhibitor serum and do I get a signal? Oh my gosh, take it off the market. No, you can't do that. You have to have good controls. And you also have to have good control serum. This is from a diagnostic company, Fadia, which is now bought by Thermo Fisher, and this is a hypothetical source. You would say that the number of people who are not allergenic, all of them should have no IgE binding above some threshold level. This is the amount of specific IgE. All people that are allergic, you hope, would be on this side of the curve. However, when you look at the reality of the best validated tests that are used for diagnosis, some people who are not allergic have IgE binding in that test, and some people who are truly clinically food challenge positive have essentially no detectable IgE. It's not a perfect test. That's in a truly validated model. If you're going to do serum testing, you have to make sure the serum donor has the allergy. You would like to have challenge positive subjects, but that is exceedingly rare. You want them to have objective symptoms by description, have been through a good, qualified, trusted allergist. You also need controls. You would like them to have skin prick tests to the known allergen, and you would like to have some second-party sort of validation by something like a FABIA test to show that they have IgE binding to the source material. I'm clearly not going to go through all the details of this, but basically the tests, there are many different formats that you can use. The tests are that you put the antigen on some solid phase usually, you allow the serum to bind, then you wash off unbound IgE, then you put on an anti-IgE that's labeled, that's highly specific. There are many anti-IgEs on the market that are not very specific, so be careful. If you get some binding, you may have to prove specificity using soluble inhibitors, both the test material itself as well as the potentially cross-reactive things, and you look at how specific the binding was. So EPA paid us to look at some different legumes and look for cross-reactivity. And these are complicated, but let's look at it superficially. These are Western blots of extracts of peanut and soybean and lupin and green beans and blah, 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 and 2 controls, rice and walnut. This is one soybean allergic subject. It's pretty extensively washed. This is IgE binding as detected by a very specific secondary anti-IgE. This is common bean. This is navy bean. High binding. The person's allergic to soybean. This is protein. We've shown through inhibition this is binding to the carbohydrate. This person doesn't claim to respond to beans. You look at these other bands, what the heck do they mean? Do they mean that person can't eat those? No. They show some level of IgE binding. Soybean and peanut allergic subject, similar thing. Again, we've inhibited. True peanut allergic with very little other IgE binding. These are the dominant allergens, RH2, RH6, and I'm not sure what the other one is. Oh, strong binding. Oh, what the heck's that? It's some low level of binding. It's background binding. Those proteins are probably lectins that bind IgE. How do you sort this out? Well, this is just a tip of the iceberg sort of comparison. And why we got interested in the carbohydrate was we took some peanut agglutinin, we took the alpha amylase inhibitor, we took the transgenic legume, and we took the non-transgenic legume. We did serum testing and we found, oh gosh, this guy has IgE here and here. Uh-oh, this person does too. When we did inhibition studies, we found out they weren't cross-reactive. Here's an example of that. Direct binding, this subject, if we try and inhibit with peanut agglutinin, we don't get any reduction of alpha-1 amylase inhibitor binding. If we use alpha amylase inhibitor, we get tremendous reduction in binding. If we use digested PHA, a highly glycosylated protein, it reduced it. If we use intact PHA, another bean protein, phytohemagglutinin from beans, oh, where's the binding? It's gone. That's because it's the carbohydrate. Sorry, I think being in Austria is making me nervous or something. I don't know. Maybe it's the hotbed of intense debate. I don't know. Or maybe that's me. Here's some pictures of the carbohydrates that TJ was talking about yesterday. These are plant carbohydrates, asparagine carbohydrates. If they have a alpha-1,3 fructose or a beta-1,2 xylose, some people have IgE that will bind to these. Here's another structure that can be bound from bromelain. This is from horseradish peroxidase. Those subjects, if you remove the glycans, do not bind to those proteins. If you look at, this is a source of hemocyanin, keyhole limpet, it's a crustacean. They can have glycans that can also bind IgE. If you look at insects, they can have a similar structure. If you look at fetulin from calf and you look at other human proteins, they do not have those glycans, and these people will not have IgE binding to mammalian carbohydrates. if you had some IgE binding like we showed to the alpha amylase inhibitor, you might want to use basophils as a test system to see if that IgE binding had any relevance. And the presumption is that that might be important for symptoms. I'm not going to go through the detail, and I apologize for the speed that I'm going at, but this is a peanut allergic subject, plasma from subject 728. They're highly allergic to peanut. And if you look at the Western blot, IgE Western blot, here's peanut. Tremendous binding to AirH2, AirH6, some binding to AirH1, AirH3. They also have faint binding to, oh, okay, this one is lane 3, this is navy bean. So there is some level of binding to navy bean. If we take basophils that are devoid of IgE and we put the subject's plasma on them, IgE binds to the Fc epsilon receptors on those basophils. If we then take an extract of peanut at different doses and look for the amount of histamine release, or you could look for beta-hexosaminidase at release, you see that at very low levels, this subject activates the basophils and gives you 50% release here at 0.33 nanograms per mil, and goes up to 80% at 33. When we use navy bean extract, and this is based on a total protein extract, the amount of protein, what we find is here at 10-fold, there's no release. Here at 100-fold difference, there's almost a 50% release, and it's gone here. So there's different affinity of binding, different reactivity. This says that, hey, there really is a little bit of release with this navy bean. If you look at non-transgenic P, transgenic P, so this is with alpha amylase, there's no binding, no release. And we've also done that with people who have IgE binding to the carbohydrate on alpha amylase inhibitor, and we do not see release or activation of the basophils in a cell culture assay. The interpretation, I believe, should be that that person can eat that alpha amylase inhibitor containing Cal-P or P with no harm. Because while they have IgE that binds to the carbohydrate, they undoubtedly will not have reactivity. So I don't think that there is a risk of that. Yesterday we talked a little bit about the digestibility of the proteins, and I've been involved in different studies. This one in 2004 was published, and it was a roundtable. A number of labs looked at a number of different proteins using a standard standardized enzyme digestibility assay. It is in a test tube, and it is not meant to mimic what goes on in the stomach. In my lab, we also further optimized the conditions and also put in some criteria that would say, okay, let's put a measure. These studies were based on time of disappearance of a stained band in a gel, and here we said you should put on dilutions of the target protein and be able to quantitate it, so that we're saying, let's choose the time that 90% of the protein is gone. So we said, okay, we're going to look at TJ's protein. And actually, my Ghanaian student already had some of this data. We just did it a lot better and more extensively. These are stained gels of time course of digestion in pepsin. This is pepsin in this gel and pepsin in this gel. Here we're looking at these proteins represent tender green alpha amylase inhibitor isolated from the source of the gene. These bands represent the transgenic P alpha amylase inhibitor isolated from the transgenic P. And if you look, these are two controls that don't have pepsin. Here's the time course of killed pepsin, or time 0, which represents 30 seconds of digestion, on out to 60 minutes. It's still there. The same is true of the alpha amylase from transgenic P. But we also isolated alpha amylase inhibitor from pintodine and other sources, and when we loaded it the same amount we did the digestion, it's stable in all. And guess what? That protein is in all of those beans that you guys eat without harm. So this is not a perfectly predictive test. It is a suggestion, and it may actually relate to the fact that if you had IgE binding and if it digested very rapidly, there would be lower risk of reactivity. but we're not sure. Again, it is a correlate, it's not proof of allergy. But we think that in this case, you're just the same as the native stuff with a tremendous history of safe use, and we don't think that represents extra risk. Based on a weight of evidence approach that Codex recommends and EFSA recommends, we believe that the weight of evidence indicates that transgenic alpha amylase inhibitor, as is put into cowpeas and peas and chickpeas, does not have any significant risk of allergy. We think that the strong history of safe use for this product is the best thing you should be paying attention to, because after all, what's all the rest of the stuff? There's scientific attempts to predict events. What are they trying to predict? They're trying to predict risk or not. If you have the human data, do you really need all of the animal model data and the in vitro data, et cetera, et cetera? I don't think you need it as much. You still have to go through the process. You need to know the results, because if you don't do those tests and the regulator says, oh, TJ, put it on the market, somebody else does the test, publishes it, and then they say, my gosh, the regulator didn't do their job, and TJ, you didn't do your job, so they'll beat you to death with that. So you have to do the study, you have to interpret it, you do the best job you can. I'm not saying just because I like TJ and I think this is a great product that it should go forward. I'm saying the evidence, primarily based on history of safe use, should say, hey, we don't know of a risk. And nobody, as far as I know, has been able to show that a dietary protein causes allergy just because of N-linked glycan recognition of IgE. Nor have they shown that with stability and pepsin. So let's look at this unintended effect thing, and that's kind of partly what and I'm going to be done in about 2 minutes, and then we can have questions, and you guys can beat on me like I beat on people, I'm sorry. Let's look at unintended effects. Did insertion of a gene like CP4, EPSPS, or some other gene that might be in somebody else's product actually increase risk of allergy from soybeans? Well, it's possibly, but unlikely. I think that there are a couple of things you could say. Well, did you put in a transcription factor that really boosts a lot of proteins that are allergens? Then maybe. But we know a lot about transcription factors, and the genes that have been put in right now are not. Did you insert it into the gene that's already known to be an allergen? Well, the developer has to do a lot of sequence and figure out whether it's there. And indeed, these are not. And anyway, what is the consequence? Would a 5% increase mean anything for allergy and risk? I don't think so, because you might eat twice as much bean today as you eat tomorrow, or a tenth as much today as you eat tomorrow. What's the important thing for risk? Well, if you're allergic to it, you have to avoid it. Does it matter if you avoid certain kinds of soybeans if you're allergic? No, we don't have any evidence it does. If you're allergic to soybean, you have to avoid soybean, period. So here's data actually from BASF, and I've shown this data before, so hopefully my friend in the back, who I just met yesterday, is not having a heart attack. This has been shown before. These are 1D gel blots with 2 individual sera. This one we know has soybean allergy, but they have IgE that bind to CCD, the carbohydrate. This one has soybean allergy. It does not bind to carbohydrate. If we use one-dimensional gels from reducing SDS Lenly, and we look at the non-transgenic near isoline, the transgenic, and 3 commercial varieties, we see a difference, but guess what? That's a commercial variety. And this one is carbohydrate. If we use non-reducing gels, we see different patterns. Why is that? Because in this condition, we reduce the disulfide bonds that help hold proteins together as subunits, or they can unfold proteins that are linked. If you have non-reducing, they run more as the whole integrated length. You see different patterns. If you look at this subject, you see different patterns as well, but you don't see that same binding band that you had here. We put purified IgE at different concentrations to give us a hint of how much we're actually developing exposure. But EFSA is interested also in very detailed component resolve sort of analysis. So for this we use 2D gel blocks and looked at the isoline, the transgenic, the commercial varieties. I'm showing you the CCD binding subject, and here's the CCD protein. And you could, we did not, but we could have identified it by MSMS, but we didn't do that. Is there a risk? Well, guess what? It's in the commercial variety. So what happens, though, if it was here and not here? What would the regulator say? Oh my God, what are you going to do? Now you have to go to great lengths. Is there a risk? That's in commercial varieties. So it's no different in risk than we have in the market today. Anyway, there's a ton of data that we're not showing. Hopefully we'll be publishing it soon. That was presented at the food allergy meeting in Venice last winter. Risk of allergy for soybeans. Somebody with soybean allergy must avoid soybeans to remain symptom-free. We do not, in the marketplace today, in the industry today, have a process for selecting non-GM commercial varieties based on potential allergenicity. We also, though, have to take this data and consider, what about PMM? What about risk to subjects? We don't eat soybeans. Here's what we eat, right? Soybeans are some % of composition of all these products. A lot of them are soybean oil, which has essentially no protein because it's hexane purified. But what is the amount of soybean in all these different products? If you label it GM, what does that tell you about risk? I'm not going to answer that. I think you need to think about it. I need to think about it. And then we need to say, is it important? Could we monitor? And if somebody develops allergy 10 years from now, can we say it's because Roundup Ready soybeans is in one of these products at 0.1%? Think about that, please. Okay, I want to just mention there are unproven tests that people are using. I've done a lot of testing with animal models, and from the data I've seen, we do not yet have a validated animal model to predict allergenicity, but there will be presentations this afternoon, and maybe they've made some strides. Heat stability, it's unclear what should be measured because depending on what you measure, proteins don't just disappear with heating. They're still there. They may unfold. Targeted serum testing, that means you take serum from a lot of people and you test it. There's no proof that it's predictive. There's a lot of false positive tests. Active post-market surveillance, I'm not sure that we can do it in a meaningful way. T-cell activation, we do not know enough about T-cell activation to say it's useful in the risk assessment. But we can argue about that at lunch, please. I would rather say, okay, here's my summary. History of safe use is the most important thing. Bioinformatics is clearly important, but there are shades of gray. If you have to do serum testing, you have to do it well, and you need people who are very experienced at that. The example of GM cowpeas, I think, suggests that there's no real risk due to potential cross-reactivity. I don't think you should have to do serum testing. I have many people to thank, including a fool who's from Ghana. She's gone. Here's a postdoc that was with me, Steve Taylor. A number of people, including the companies that support Allergen Online, they don't get a vote on what's in there. They give money, and they can say who's an expert on the panel, but then every year we release it whether they like it or not. It's there. I've done studies for the US government and for individual companies as well. Okay, sorry that's a little bit long. It was very fast. You can't have a PDF of this. But I would be happy for you to beat our meat with your questions, and I'll do my best to answer.